and we'd like to welcome everyone to the session today. This session will be recorded, just to let you know, so that we can put it back up on our website for later use. So just letting you know that the recording and the questions later on will be recorded. So just to also, if you've got any, if you want to contact us in the future about the future webinar series, by all means do that through our Twitter group or LinkedIn as well. So without further ado, I'd like to introduce Edgar Jacoby from Janssen. And today's talk will be focusing on target validation of cellular phenotypic screens, knowing the knowns. And we hope that you enjoy this talk, and we'll also be having a panel session after Edgar's talk. So if you've got questions that you think of during the talk that you'd like to put to a panel of experienced life science informaticians and drug discoverer people, then by all means put those in the question part of the GoToWebinar. But I'll just hand us over to Edgar, and I'll just briefly introduce him as we sort out switching presenters. So Edgar is now at Janssen, and before that was working at Novartis in their research group, and Edgar was one of the sort of key groups within Open PHACTS in terms of the business or the life science contribution to Open PHACTS. So much of this work that perhaps is presented here today is going to be done focusing on the work that Edgar's been doing over the last few years, and some of the papers that have been published, which I'm sure he'll mention too. So just
thank you, Nick. Do you see my screen now? Yeah, show my screen. Do you see the screen?
Yes.
Okay, I put it on full mode. Okay, so the title today is Open PHACTS Computational Protocols for In Silico Target Validation in Cellular Phenotypic Screening, Knowing the Knowns. And its work, as Nick already presented, done in the Open PHACTS Consortium. And I want to acknowledge a couple of people before starting, Jean-Marc and Herman here in Janssen, who helped to implement the pipeline pilot protocols, Daniela and Barbara in Gerhard's group, who worked implementing basically the same protocols in NIME, and Christian and Andre also helped with pipeline pilot to get the protocols and all the nodes available in pipeline pilot. So I will talk about objectives of this study and come then to 6 computational protocols we agreed on, and apply this then to a phenotypic screen we have been interested in, and it's a published screen, and then have some conclusions. So in phenotypic screening, I think it's really a must that you should know what is known about your compound. So knowing the knowns is a must in phenotypic screening. And these screens are typically agnostic of efficacy targets in the beginning, and still the objective is to understand the molecular mechanism which needs to be elucidated and validated. And here in silico assessment, knowing the knowns is a key step. And with the Open PHACTS software and databases, I think we have an integration which allows to do such analyses. And the objective is then to enable insightful interpretation of these screens, and in context of the up-to-date established knowledge in drug discovery. So we had a workshop in Santiago de Compostela in 2015, and there we had a very good discussion about phenotypic screening and target validation, and we came up with a many ideas, and among these ideas were 6 protocols we said we would implement as a deliverable of the Open PHACTS project. The first thing is to know what classification, what target classifications or compound classifications your compound belong to. If you have hits, let's say you have a phenotypic screen and you have a few hundreds of hits, and you want to know for each of them, is it a kinase inhibitor, is it a known GPCR inhibitor, at what potency level is it active on these possible efficacy targets. Then if you have these possible targets, you want to know more about these targets via the compound link. You want to know the Go annotation, meaning the cellular components these compounds are hitting via these targets, the function and the cellular processes which you have in the Go annotation. You want to know possible target pathways, so which you have in the wiki pathways components of Open PHACTS. For example, are multiple compounds via multiple targets hitting the same pathway? This would be a strong indication that this pathway has evidence in this biology you're interrogating by the phenotypic screen. Then you want to know links to disease or possible side effects. You know, typical side effects you want not to see with hit compounds are hERG side effects or other anti-targets. Are there diseases which are similar in their protein target profile than your disease of interest? We show this also here in the application. Then you want to know, are there assays, mainly biochemical or cellular assays, which show a high correlation with your phenotypic screen, so that you could potentially use this assay as a surrogate assay with a higher throughput to follow up on your phenotypic screen, or even know already the target of your phenotypic outcome. Then a 6th protocol was to identify compounds to validate or devalidate potential efficacy targets using the UFA database, which is today probably the database with the best classified and best annotated pharmacological tools we have in our hands. So these are all the probes from the Molecular Library Initiative at NIH, and also the SGC probes are in there. So these are really the best annotated compounds compared to HDS compounds which we find in Campbell. So Open PHACTS provides the required semantic integration. So here you see basically an outline of the network of URIs which are interconnected and linked data which we have in Open PHACTS. So if you have a target, you can go to the assays and the compounds. If you have a compound URI, you can navigate to compound classifications in the Campbell database. With the target URI out of Campbell, you can go into the target classifications, the GLOW classifications, the wiki pathway URIs are directly linked, and also the disease URIs from this GeneT are directly linked. So you have this type of integration possibly also in other APIs, or but here the semantic integration is the key advantage, which allows you to use synonyms without having to care about which synonym you actually have used in your queries. So the protocols 1 to 4 follow a quite generic pipelining flow chart. It is that you start with a URI list of your active compound from your phenotypic screen, and then you look into the compound classification of CABI, and you do some data aggregation, and you have an output list. On the other hand, you go look into compound classification in Campbell. The compound pharmacology is retrieved with the compound pharmacology list call. You apply some filters on the p-Campbell value, which is a generalized potency value. You filter on the target type. So we in the 2.0 implementation, we use the Campbell confidence scores, target confidence scores 9, which is that the target is really characterized at the molecular level for specific proteins. And then you look into target classifications, Campbell and Go, do again data aggregation and some statistics to count, for example, the number of actives, the number of unique compounds, or the number of targets, and things like that, very essential statistics, and provide an output list. The same you can do with pathways for targets, with the call which is existing in the API and which queries the wiki pathways data, and you can do it with disease for target lists calls which query the DisGenet data. So in Pipeline Pilot, this looks like the following, the protocol 1. So you go in with a compound list, which you query. I show it later how you get the compound list. And you pull the compounds in a cache. So Pipeline Pilot has an advantage, I think, to allow caching more directly than other tools, and make then use of these caches to join the data more easily. So then you retrieve for the compounds the compound pharmacology list. You put again the pharmacology list, meaning the targets, the compound interaction file, into the cache. And then you look for each target, the target classification up, and you put again this classification into a cache. What you then do in step 2 here in this part of the data stream, you basically do the data joining in the inverse order. So you start with the target classification, you join the targets, and you join the compounds to the targets. And then you can have an unmerged output list here in step 3, where basically you have a stream of target classification, target, compound, and activity values for each reported activity. Or you can do some basic statistics, some aggregation statistics, where you count the number of unique activities. So sometimes you have the same activity values reported in different assays in Campbell, because different publications basically report the same data, and these are different activities for Campbell, the same activity, but different activity reports in Campbell. You count the number of unique compounds, and you count the number of unique targets, and this is then put into an output table, a merged output table. And this is what you report then as output. I will show this maybe also later at the end. Then the correlation robot is a protocol which in Pipeline Pilot is extremely simple to implement. The first thing you do, you pull all kinases from the kinase classification tree, from the target classification tree of Campbell. So you give the ID number of kinases, and this first data stream pulls all kinases for which there is data in Campbell. Then here you pull in the second data stream, you pull the phenotypic assay data and put it into a cache. And then you do the following. You read each kinase. For each kinase, you pull all target pharmacology via the API call, and you get for that kinase all assays. And then you group by assay. So for each assay, for each kinase, you do basically the correlation of your phenotypic data with that particular assay you have just pulled. Here in this step, we do at the moment a very simple correlation analysis, in the sense that we only look at the compounds which are common above a given threshold. We could do more sophisticated rank order correlations or Pearson correlations, or even more sophisticated statistical analysis at this step. But at the moment we have not done, for the simple reason that the data matrices we have in Campbell are sparse, and so using corporate data where we have full HDS hit lists, this makes more sense to have more advanced statistical correlation analyses. And then again, you put this into an output table. So it's extremely simple to have a correlation robot, and you can correlate hundreds of assays with your phenotypic assay. And then here the UFA query is extremely simple. Again, you simply read the UFA interaction file, which is deposited at the UFA website. You have to select the data by taking affinity values which are the most appropriate, and this is done here in these steps. Most appropriate means that you have to make a selection, the maximum value or the median value, depending on whether these values are given, if there are multiple activities reported for a given compound in the UFA interaction file. Then in the UFA interaction file you have the UniProt IDs, and the next step is then of course for the list of targets you have extracted for your candidate compounds. You also get the UFA, the UniProt ID, and then basically you have everything together. You do a join on the UniProt ID here in this step, and make sure that you distinguish antagonist from agonist, from other mode of actions, from other modalities, and then basically write an output file where you have the interactions you get for each target you had proposed as candidate compounds if they exist in the UFA interaction file. So now let's apply this to the layman AC splicing assay. So the aim here in this assay, which was done at NIH, is to find splicing correctors for premature aging disease, which is caused by a mutation which opens in the layman AC gene an alternative cryptic splicing site, and the consequence of this is that the protein is accumulating, the non-correctly processed protein is accumulating, and this has a couple of effects, negative effects on cellular functions, and results in the disease at the end. So these are typical examples of patients and children who have this premature aging. You see this sometimes, and here people are looking for splicing correctors. So in the PubChem bioassay, you find an assay which is looking for correction of spliced protein, and there are around 280 bioactives in that assay reported, which potencies up to 35 nanomolar, micromolar, sorry, and there are 85 chemically diverse compounds with P Campbell values higher than 6, and these were the compounds we identified and analyzed here in more detail. So what does the annotation tell us? So if you look at the Campbell annotation, there are around 47 kinases which show up for 8 compounds on 27 targets, so 47 kinase activities, so report activities in Campbell on 27 kinases with 8 compounds, and among them are, for example, DYRK1A inhibitors, which are reported in literature to modulate alternative messenger RNA splicing. There are GPCRs which come up with 14 compounds on 19 targets, and most prominent are monoamine receptor activities, and there are also epigenetic regulators with 17 compounds on 5 targets. Then Go annotations, there are 186 Go components which are found. 13 compounds are directly linked to the spliceosomal complex via heterogeneous nuclear ribonucleoprotein A1 and to survival motor neuron protein. there are around 1,300 Go process terms found, and many of them are linked to DNA-related processes via Bloom protein. Again, 13 compounds are linked to the spliceosomal complex, and 340 Go terms are found. So you see this explosion of terms when you have suddenly one protein linked to multiple Go terms, and there's further analysis needed to really find the relevant ones in that data. Then the kinase correlation robot pointed us to the ERK2 kinase, which has 8 compounds in common. So here we would recommend to test further ERK2 compounds and to get a better coverage. The DisGenet annotation points us to 3,600 diseases and side effects. Around 90 have more than 20 potential efficacy target linked. So there are many kinases again which link to various neoplasm cancers, and then spinal muscular atrophy, which is probably the most relevant one here as a direct link via the survival motor protein, is also highlighted. So in here it really requires more expertise in biology and special disease biology to recognize the relevant links to the observed phenotype. Jacoby is pointing with 5 compounds to include 5 metabolites and anti-neoplastic agents, counts with more than 5 compounds, and these include fluorouracil, camptothecin, and rotenone. And rotenone is a compound in the literature discussed to modulate splicing of several genes. Then the UFA box analyses suggest to test 79 compounds to test 86 of the 105 potential targets. And again, very prominent here are monoamine receptor ligands and kinase inhibitors. So this, of course, has to be taken with a grain of salt. We have not analyzed negatives in this analysis. We have only looked at positive hits. Campbell is reporting positive activities, negatives in that sense not included in the data, and more to rough statistical analysis is needed to really find those targets and finally which distinguish the noise from the signal from the noise, sorry. So in conclusion, Open PHACTS provides a seamless integration for analysis of phenotypic screening data. The interplay of Open PHACTS API calls with data pipelining and analysis tools allow great flexibility. We have shown this with Pipeline Pilot, we have shown this with KNIME. We have shown that we can integrate data from UFA, which is not integrated in Open PHACTS at the moment, to full extent. And I think with this we can show that knowing the knowns is possible for everyone. And here I stop with presenting.
Thanks very much for that, Edgar. Are you okay to take a couple of questions?
Yeah, I'm fine, yeah.
Okay, sure. And equally, let me also introduce Andre Caracotti, who is also part of our panel for this afternoon. Andre works for Biovia and has been instrumental in setting up some of the Pipeline Pilot workflows, not just for Edgar's talk, but for Open PHACTS in general. So we welcome you, Andre, to our panel this afternoon. We've had a couple of questions from the audience already that they've typed in, so I'll just start with a couple of general ones maybe to start with, and then we can see where this takes us. So Edgar, just a quick comment about is there any differences in this sort of phenotypic validation compared to perhaps more traditional target validation? Did you see, was there anything that you had to take special care of or such?
Yeah, I think we learned that we need the negative data to really distinguish the true targets. There might be, for example, the biogenic amine receptors, which we found here in the SID list. Some of these receptors are quite promiscuous, and it will be possible that in the inactive compounds, which we didn't analyze because we didn't have the inactives from that screen, that there are equally number of biogenic amine receptor targets in the inactives versus the actives, and that would speak against these. So I think what I really learned is that it's necessary to include the inactive data, and this was also one of the comments from one of the referees. We submitted this as a paper and to MedChem Communications, which is now accepted, but I think the learning is that we have to, for phenotypic screening, we have to look into the inactives to do really in silico target assessment.
Okay. Andre, maybe just posing that question to you. In terms of the workflow that you've built in Pipeline Pilot, was there anything you needed to be, you had to do particularly for the phenotypic assays, or was it something that you built before?
So just to clarify my role in the Open PHACTS work, I've continued some work that had been started previously at Excelris, now Biovia, in building a component collection that wraps basically every API call that's available in the Open PHACTS API into a Pipeline Pilot component. So we have specialized components for each API call in the API, in the Open PHACTS API. And we now have a collection that we try to update with every version of the API. And that's the collection that Edgar installed on his Pipeline Pilot server and built these protocols with. I didn't have any special role in building the protocols that Edgar has talked about, other than, again, the fact that he has used the components that I created. So I don't really have any input in that particular work, other than to say that, with these components, it was probably a little easier than using Pipeline Pilot's, low-level components like the HTTP connector that can connect to an API endpoint and do something with it. So I think the components facilitate building these protocols. But again, I didn't have anything to do with these particular protocols.
Okay, sorry for the confusion. We've had a question about, Edgar, you mentioned about the splice variant target, I think, earlier on in your talk. How were you able to specify that?
We took the assay as such it is deposited and used this as an example for these protocols to illustrate how we would annotate phenotypic screens in the future. So this assay here is looking for splice correctors and reads the readout is correctly spliced protein, and this is the readout of the assay, if this is the question.
Okay. Well, I'm sure we can see if that answered Chris's question. Jeremy has asked us a question about, would this workflow be able to detect when phenotypic assays involve multiple targets?
Yes. So if these were known targets, we would see annotations of a compound to multiple targets, yes.
Okay. And just perhaps a general question about the efficiency of this. What do you feel in terms of the efficiency versus the quality boundaries in this type of approach? Were there anything that you've learned from this approach on the sort of the efficiency of running it in terms of speed and also quality?
Yeah, I think what is helping a lot is that for every API call we have now a component in Pipeline Pilot, and the flatten tools to flatten the data hierarchy which are available in Pipeline Pilot make it very efficient to program this. You know, I think the protocols take to run 5-10 minutes each, and we could still improve the query efficiency and speed by working probably not on the protocols themselves, but on working on the server. Maybe also some API calls could be refined, but I think practically these protocols are usable, and so we plan to integrate our own in-house data and have then these type of protocols applied to you know, the whole benefit will come from in-house data where we have more complete data matrices, HDS, H lists, and including inactives, especially for phenotypic screens. I think this is really necessary to cut down this explosion of data when you go to these terms, go terms and diseases. You have suddenly there's too much data, and you have to make sense out of that data. So you could analyze similarity between these terms in terms of proteins they integrate, but there are many other possibilities to make here further progress, and so there is still some work to do there.
Okay, thanks for that. Ian's asked us about, I mean, maybe, I know we've got Jean-Marc also on the panel, and Bryn. Were there any surprises in this work that you found that you weren't expecting? Maybe this is to Jean-Marc first. So welcome, Jean-Marc. I know you're a colleague of Edgar's at Janssen. In terms of the work that you were doing on the phenotypic workflows, was there anything that was a surprise? Are you on mute, Jean-Marc? Okay, maybe whilst Jean-Marc sorts out audio. Edgar, just maybe turning that question back to you. Was there anything that you found as a surprise during this work?
Yeah, indeed, there were a couple of compounds which there is already some knowledge about these compounds, and especially in this biology. And being new to that biology, I found it quite useful to be pointed to such references, and which I was not aware of before.
Okay. Just I think just going back to your point about some of the Go terms, and Peter's asked us a question about how will you tackle reducing the amount of Go terms retrieved. Have you got any thoughts on that?
Yeah, the first thing I think will be to use in future inactive data, and I think this is also a requirement then to Campbell and the data providers to store this negative data. This will allow really to trim the list down, so targets which are suggested by compounds which are indeed inactives in a phenotypic screen and which are also suggested by actives cannot be true targets, because otherwise they would not be in the inactive list. And so this is one step. The other step is really trying to find similarities among these Go terms. You know, they are hierarchical and they depend on each other, and so there might be to have groups of Go terms which belong together. I think the clustering analysis of the Go terms and their similarity in the disease terms will shrink the number to more meaningful terms. because otherwise you really overfloat it with 3,600 disease terms, and you don't know where to look. Of course, you look for what you know already, but this is not objective. It's really to give you new insights. You know, here we see that there are some other splicing diseases which are related to this biology here, but there is also, of course, then, you know, what's really the challenge is that you have to have really an interdisciplinary team on board to analyze all this data. You know, we did this from the cheminformatics, bioinformatics perspective, and just first putting these pipelines together. But really, to make sense of it, you need biologists who know the biology they want to study in great detail, and then they can make sense out of these terms. Still, we need further work to shrink these numbers of terms into more meaningful groups of terms which belong together.
Okay, no, that makes sense. Peter's also asked a question about integrating in-house data. Integrating in-house data with this, and I know that, just as a bit of a plug, our next Open PHACTS webinar actually will be including a bit more on how people are using Open PHACTS with bringing together public and private data. But Edgar or Jean-Marc, did you have any thoughts on what you've done already with those types of integration?
Jean-Marc, you can take that.
Sorry, I was just out. Sorry, I missed the question.
It's okay. There was a question about integrating in-house data with what Edgar was describing around the public data here with phenotypic targets. So, was there anything you wanted to mention about that before our session next month? Any learnings or?
The crux there is you can link or integrate at different levels. So let's say that the simplest level is with pipeline pilots. You can do some analysis for the public data and Open PHACTS data, and you can kind of mirror that with the necessary changes for internal data. This is how we, for example, do analysis on compound identification for different targets. So based on the target information in Open PHACTS, we get compounds, and based on target information in our database, we also get compounds. And then we try to reconcile the compound structures must be called the same, and then the activity data must be called the same, and the target must be called the same, and the description must be called the same. So there's a lot of like matching that you can do without touching too much your internal data. It works, but the disadvantage is that it doesn't work for everybody. You have to know in pipeline pilot more or less what you're doing, and it's not extremely fast. With extremely fast, I would mean having the answer within a few seconds. If you have to search for activity data over millions of records in our own internal database with pipeline pilot, it can take 15 to 20 minutes. So that's still not extravagantly long, but for chemists waiting to see something happens on the screen, that is long. That's the first level, and that really works. It's been working very well. We have developed scripts like that. But for the end users, let's say the chemists, they would need something that Open PHACTS has done, and that is linking the data at the source. So you need to transform your data into semantic format, or at least allow calls to your database in a certain form with the results being in semantic format. so that you can link the data semantically with Open PHACTS or other semantically formatted public data. And that's what we're working on, and that's what I can talk about next time. So I won't give too many details there, but instead of doing an ad hoc mapping every time you do a query, the ad hoc mapping is done beforehand with in-house and public data. So end users like chemists can do one query and get all the results in one query, in one end result, in one worksheet, let's say, of applications they know. That's the two main levels how you can put public and private data together. But it's not it is fairly simple in theory, but in practice it is quite demanding on transforming relational data into a semantically acceptable format. So you need to think in a different way.
Thanks for that, Jean-Marc. And as you say, our next webinar will be covering some of those details in the end of May. So just trying to, there's a couple more questions to look at perhaps before we want to bring this to a close. So, I mean, it's actually quite a broad question that Peter's asked about visualization, and maybe it's a talk in itself, but was there anything that you needed to think about in terms of the visualization of the data that was particularly specific to this, or were you able to use existing visualization tools?
Yeah, so we used the 3DX Explorer, which is a Janssen internal tool to write out the data into, and so for the protocols which you make available, we give it into an Excel sheet, and but in-house we have a visualization tool of the data analysis and visualization software, which allows us to do things afterwards in a comprehensive and integrated manner. But this we cannot share, of course.
But maybe that's almost a talk in its own right, in terms of perhaps not necessarily the internal tools, but perhaps this community thinking about what are good ways of visualizing these types of output. I know many people will be using the sort of standard toolkit of sort of analytics tools in their companies or in their work. So it maybe would be interesting to see if anyone wanted to do a particular talk on that in the coming months. So maybe that's perhaps quite a good time to bring this to a close. We'd certainly like to thank Edgar and our other panelists, Andre and Jean-Marc. I'd just like to briefly mention our next webinar that's coming up at the end of May. This is going to be the one on, as we've just mentioned, integrating data with Open PHACTS, private and public. And we have two speakers at that session. Jean-Marc, who you briefly heard already as part of this work on target validation, and also Yusa Bartek, who's at the University of Strathclyde and did a lot of that work during a placement at GSK. But it just leaves me to thank all our speakers this afternoon, and thank you for your questions. If you've got any other questions on this topic, then by all means get in touch, and we'll be uploading the talk and the slides to SlideShare in the coming days. And equally to wish you a good rest of the week, and thank you for joining.